where you have a lot of these really short paths, and that facilitates local information processing. And then you have occasionally these longer distance connections that could, in theory, facilitate global information transition across the rest of the brain. So if these were the only two things that were going on, if these are the only two features that are conserved, then these long distance connections could kind of connect any two regions in the brain. They don't have to connect anything specific. So the next question that we wanted to ask is if these long distance connections are random or if they are preferentially connecting similar areas. If I can advance the slide, I will tell you what the answer is. Back button. Aim it up. This is like really suspenseful. I know you guys really want to know if these connections are random or not. It's just... Sorry. We might have to like turn it off and turn it on again. It might help. Should I just like keep talking? Or? I guess, yeah. Okay. Well, maybe I'll give you a little preview. The answer is they're not placed randomly. They preferentially connect specific brain regions. So, you know, new cliffhanger. What brain region? Oh. Ah, great. Yes, they are not random. Yep, amazing. And this is another feature that is conserved across evolutionary scales. And we can do this basically by testing against a null model where the long distance connections are random. And it turns out that these long distance connections are sort of preferentially connecting these really tightly linked modules. So we have tightly linked modules in the brain. These are giving us local information integration. And we then have these long-distance connections that link the modules together. That allows us to segregate information when we need to, but then communicate it really quickly later on, which gives the brain, theoretically, a really wide array of sort of functions. So in summary, some of the simple organizational principles are spatial, and they're consistent across systems. Brains have many short-distance connections and a few long-distance connections. As I'm sure you will all remember now, those are not placed randomly. So we're going to change gears pretty drastically now from sort of this just description of what the connections in the brain look like to a formal model for how this structure constrains activity propagation and how that's influenced by the topology. So once again, to reiterate, what we have, what I just talked about, is this network of structural links. But what we want is to know how activity in the brain spreads along those structural links. And importantly, we want this theory to be predictive. So really what we want is a way to know if we change activity in one region of the brain, what is going to happen everywhere else? How is that going to influence the rest of the activity? And the candidate model that we're using comes from network control theory. Given that neural processes can be approximated by linearized generalizations of nonlinear models, we can then use network control, which essentially stipulates a linear model of how dynamic activity kind of propagates along a complex network system. And this is the model up here. It incorporates the state of the brain in a vector x. So this is your activity. Whenever I'm talking about brain states, think just pattern of activity. It could be bold, it could be ephys, it could be whatever. It also incorporates the weighted adjacency matrix that I spent a lot of time talking about in the first section. And importantly, it has this third component that is sort of the input. So B here is it's called the control set. This is telling you which regions you want to change. And U is the control energy, which is essentially how much you're changing them. And this energy is going to come back a lot in the talk. I'm going to talk about it quite frequently. And essentially what we are going to do is we're going to interpret control setups that use less energy as sort of being more efficient. And then we're going to use that to see if we can learn things about the brain. Okay, so you can, one way to represent kind of all the different patterns of brain activity is with something called an energy landscape, which I show over here. And basically, every brane state falls somewhere on that landscape, and some of them kind of require more energy to get to than others. So given we can think about the states this way, we want to ask if the pattern of connections at a given node sort of affects how it will move the brane through this energy landscape. And we're going to talk about two different strategies in this talk. One of them is average controllability. The intuition behind this is that a node that's a good average controller would go to a lot of easily reachable states compared to modal controllability, which is kind of the opposite. It would steer to a few more difficult to reach states. Mathematically, average controllability is based off of the T-steps controllability Gramian, which is given up here by W. Really the only important thing you need to know about the Gramian is that it incorporates A, which is your structural connectivity, and B, which is the regions you want to control. So those are the things that it depends on. And mathematically, this is the same as kind of the average energy to reach all possible states. It's also the area under the curve of the impulse response to your system, if you like to think about impulse responses. Modal controllability is a little bit different. Modal controllability depends on the eigen decomposition of A, your structural connectivity. So, you break up A into eigenvalues lambda and eigenvectors V. The modal controllability of node I is basically going to be a sum of the ith entry of every eigenvector, weighted by the eigenvalue, but it's weighted such that smaller eigenvalues have more of a contribution to the metric. So it's weighted towards the difficult to control smaller modes of the system. And the way that I like to think about modal controllability is that if you add like a little bit of energy to a node that's a good modal controller, it'll result in a really big perturbation to all of the modes of the system, rather than just the big ones. So maybe that gives you some intuition for these metrics, but do they differ in meaningful ways across different things that we're interested in neuroscience? So first of all, I want to point out that these metrics do differ across null models, so they do have enough variability in that sense. On this plot, we have modal controllability on the y-axis and average on the x-axis. All the colors are just kind of benchmark models really common in network science, and you can just see that they kind of fall into different spaces on this plot. But importantly, these also differ across brain regions. So regions that are kind of known to affect control of cognition, which you could intuitively think of as maybe a more difficult to reach brain state, are really high in modal controllability. And this is contrast with regions that are active at rest, which you could think of as maybe more nearby states that will be really high in average controllability. And lastly, these metrics also differ across age and cognitive ability. So on average, for older populations of youth, they will have higher scores for both modal and average controllability. And additionally, youth with better scores on these metrics will tend to perform better on cognitive tasks. And so if we take all of this together, we are saying that this suggests that this theory could be a useful marker of how the brain enacts control to change network function. But what about the topology? Once again, the first talk was all about topology of the brain. How does that affect these kinds of measures? And so we wanted to get really an analytical understanding of how the topology affects this. And so we're going to do this with kind of a toy model of a network shown up here in the corner. So all of the red circles are called driver nodes. Those are the ones that you're changing, that you're adding input into. And all of the blue ones are non-driver nodes. Those are the ones that you're kind of observing how their function changes. And you can gain an analytical understanding of how easy it is to control this network based only on the directed connections between the red and blue nodes. So you can simplify it a little bit in this specific setup. And the take-home is that the dissimilarly distributed edge weights in this simplified network decreases the energy required for control. And what we mean by dissimilarly distributed is basically the vectors that make up the edge weights onto each non-driver node. So if you look in this toy example, this first blue node has a bunch of pink vectors coming into it, so you would have one pink vector of inputs onto that blue node. You also have a yellow vector and a gray vector, and you want those vectors to be different if you want easy control. I'm going to walk you through an example of it because it's a bit hard to grasp like this. So even more simplified model, we have independent inputs onto our two non-driver nodes. So intuitively, this network would be really easy to control. If you want to change the light blue node, all you do is add energy to the lighter red nodes, and you can change it differentially from the dark blue node. And it's really easy to do that because these inputs are independent. And then because the inputs are independent, when you plot those input vectors in a three-dimensional space, they're going to be orthogonal. And because they're orthogonal, you'll be able to draw a really big parallelotope between those vectors. And so the geometric intuition that you can take home is that the edge weights that maximize the volume of this parallelotope are also going to make control easier. So one more example to try and get a hang of this. This situation is kind of the opposite. The inputs onto the two non-driver nodes are very, very similar. The only difference is this really weak brown connection in the bottom. So if you want to differentially change anything, it all has to come from there. Because they're similar, you plot them in this space, they make a very small parallelotope, and this network would be very difficult to control and require a lot of energy. So how does this sort of parallelotope size stack up, once again, across evolutionary scales? And what we find is that the sizes of these parallelotopes, kind of relative to the strength of the network, is optimized as you go through evolutionary scales. So it's most optimized in humans, followed by mice, and then followed by Drosophila. Okay, so quick summary of, oh no, not again. Okay, good. Quick summary. Network control theory stipulates a model of how activity is constrained to propagate along white matter tracks. We think that it could be a useful marker for how the brain enacts control to change function. And topology, specifically the diversity of these input edges, influences the ease of control. Okay, so the last thing I want to talk to you about are potential uses of this model in designing stimulation therapies. So generally, when you're thinking about a stimulation therapy, you know kind of what the brain looks like in its pathological state, and you know how you want to change it. So there's really a specific target state that you want to get to. And thus far, I really haven't talked about any specific target states. I've sort of just talked about states in these really broad classes. So we need to update our model a little bit. So what we want is, for finite time, finite input or energy, for a specified initial and target state, and given this model of dynamics, we want to know how easy is it to get to our target state, and can we make it any easier? And we can answer this question with an optimal control model. This is essentially just a cost function that is minimizing the input and the distance traveled away from your target state for a given control setup. So you can compare different networks, different control sets, and different initial and target states to find out which transitions are going to be easier, and then the idea is that those would be better candidates for stimulation. So we are going to test this model in a really unique data set that we have. So we first of all have electrocorticography data from humans. This is an invasive way of directly recording neural activity that is paired with a stimulation regimen. Once again, I apologize for the text kind of covering that. The orange box says stim above it, so they're stimulating during that period. And then we're going to pair this with an estimate of cognition that we can get in real time. So essentially, this is just a classifier trained to recognize cognitive performance based on their neural activity. And we put these together, we get the white matter network, the brain activity, the cognition, before and after a really extensive stimulation paradigm. What we want to know is how the white matter network architecture guides direct electrical stimulation through these optimal state transitions from where the brain was initially to some sort of idealized good memory state that we can get with the classifier. And what we find is that when you're enacting these transitions to this specific target state, the energy depends, first of all, on the distance you're traversing in state space, which I think is pretty intuitive. If your initial and target distance is very large, so those vectors are just very far apart, the model predicts that's going to be more difficult to stimulate to get there. But importantly, it also depends on your cognitive state, independent of this distance in state space. So the model also predicts that if initially you are not very likely to encode memories, you're in a poor memory state, it's going to take a lot more energy to get you to that target state than if you were already doing pretty well. And then if we combine this estimate of cognition with some information about the network topology and the topology of the stimulated node, then we can basically explain a lot of the variance in the energy for this model. So kind of in addition to predicting which stimulation paradigms would be effective, we also want to know if we can take any steps towards explaining the observed outcome of stimulation. So we also wanted to see if the modal controllability of the region stimulated did anything to predict the change in cognition resulting from it. And generally speaking, when you stimulate regions in the brain that have much higher modal controllability, you will tend to have a larger cognitive improvements resulting from that stimulation. Okay, so in summary, we use some empirical stimulation aimed at improving memory to show, some preliminary evidence that these metrics could predict cognitive outcomes. And we talked about a model, an optimal control model, that incorporates the current cognitive state as well as the white matter architecture that could potentially tell us about the efficacy of a given stimulation paradigm. Formatting on this one's weird too, but very broad summary of what I talked about, the work in our lab, trying to quantify the incredibly rich network architecture of the brain. I then talked about a specific model for how that architecture constrains the propagation of activity, and the first steps toward applying network control for optimizing stimulation. And then with that, I want to thank everyone in our lab currently, as well as a lot of the people who have now left to become faculty members who are important in this, and Danny. I guess Danny isn't on here. And then also all of our funding. And I'd like to thank all of you for listening. And then with that, I'm going to introduce our next speaker, Pamela Douglas.